when you started out, it wasn't clear how CRISPR-Cas9 was operating, and I think many people thought it would likely be working on RNA the way some of the other bacterial immune systems operated. And then it became recognized that it's actually attacking DNA rather than RNA. And that obviously was a real game changer, because suddenly you could see how you could not only make transient use of it, you might be able to make permanent modifications in DNA. And the key question was, or one of the key questions was, could you reduce this to something that you could make work in not just bacteria, but in other cell types? And you really made the key contribution in showing what the minimal system would be. I'd love to have you tell everyone about how you came to this.
Well, I think what makes CRISPR so interesting, both as a biological phenomenon and as a technology, is that it's a seek and destroy sort of mechanism in biology that could be harnessed for a different purpose as a technology. And to describe that, I guess I would start by saying that it's a programmable system. So it's a way that bacteria can learn about the genetic material of viruses that infect them. And not only that, to store a genetic vaccination card in their DNA that allows them to essentially remember what viruses have infected them in the past, and not only remember them, but also find them and cut them up if they should show up again. And that was what was so exciting and so interesting, was to discover that this system is truly programmable, and that cells are using it to, in real time, acquire immunity to viruses. And this is happening in bacteria. And so by understanding and really digging into the chemistry of that and understanding the molecular basis for that process, we recognized, and we here as the royal we, it's myself and Emmanuel, but as well as our lab members who were studying this process, we recognized that it could be utilized as a tool to change the DNA sequence in any kind of cell.
So in 2012, you published this remarkable paper in which you took biochemically purified components of the CRISPR system and in a test tube demonstrated that these enzymes could do the cutting of foreign DNA that was targeted by a specific RNA molecule for the first time. And that really proved that we understood what the elements were, and you showed that it in fact could be programmed with any desired piece of RNA. That must have been an amazing experience.
It was really exciting. I mean, that's, you and I both know, Rick, this is what we live for in science, right? Is those aha moments, and they don't come along every day, but when they do, it's very, very exciting, because you feel like you're understanding something about our planet and life on our planet for the very first time. And that was the case for us with this work on CRISPR, was not only the fascinating biological pathway that we were studying, but also the recognition that we could now, with our knowledge, use that to harness it as a powerful tool to manipulate DNA in any kind of cell.
So tell us about the course of your own thinking and applications, as well as the rest of the field, because this obviously very rapidly was a tidal wave that overtook all of molecular biology, and now is starting to find its way not just theoretically toward the clinic, but with FDA-approved drugs.
Yeah, no, it's amazing. While you were asking the question, I was actually thinking about a dinner I had with my neighbor right around that time in those, early days. It was actually before we had published that paper in 2012, but when we knew about how the CRISPR system was working. And my neighbor is a computer scientist. He's not a biologist, but he was over for dinner with his kids, and our kids were playing and we were chatting. And he said, "So what's going on in the lab?" And I said, "Oh my gosh, Bob, I have to tell you about this unbelievable thing we're doing that's just blowing my mind. It's so interesting." And he got it immediately because he's a programmer. And so he said, oh, you mean you can program this thing to go into a cell and look up a particular DNA sequence and trigger a change? And I said, that's right. And he said, wow, that is going to be absolutely huge. And he was right. He was right. So it was the kind of discovery that immediately, I think everybody who heard about it or understood it could understand the implications. And so what's happened over the last now more than a decade since that work was originally published is that, many labs globally have adopted the CRISPR technology for all kinds of fascinating applications, not only to make fundamental discoveries about genetics, but also to, as you just said, to manipulate DNA sequences, even in patients, in humans that have a genetic disorder where a targeted change to their DNA can be curative. And so back in December of 2023, so just a couple of months ago, we saw approval from the Food and Drug Administration here in the United States, as well as in the United Kingdom, of the first CRISPR therapeutic. It's for patients that suffer from sickle cell disease, a very well characterized genetic disorder in humans that is a great initial target for CRISPR because it's a single, very well characterized mutation in people that causes this terrible and really devastating disease. And with CRISPR now, we have an opportunity with a one and done cure that effectively creates a change in the DNA that can prevent patients from ever suffering from CRISPR, from sickle cell symptoms, again, is what it looks like.
So the example of sickle cell anemia is a fabulous example of bringing this novel technology of CRISPR gene editing to a disease that was one of the first molecularly understood human diseases. Sickle cell anemia is a disease in which copies of the beta globin gene are mutated to a form that now causes the red blood cells in the body to sickle at low oxygen concentrations, causing these vaso-occlusive crises where blood can't get through blood vessels and causes terrible pain in patients who suffer from this disease. And it historically has resulted in severe outcomes and death at young ages. And so the prospect of being able to treat this disease is really fascinating. Do you want to talk about the way that it's actually been attacked using CRISPR?
Yeah, it's a very interesting situation because, as you just said, it's a devastating disorder, very well characterized. It affects people primarily of African descent, although there's also a large population with this disease in South America, which is very interesting. So there's some fascinating, genetics going on there. And at least in some of these patient populations, these are populations that haven't been very well treated in the past by the medical community. So I'm really proud that CRISPR is coming to bear on a disease that affects them right now and can in fact have a very positive outcome. That being said, it's difficult to get access to the therapy because it's treated, the way the CRISPR molecules are put into these patients is in a process that involves taking blood stem cells. These are cells that can give rise to mature adult red blood cells, taking those from the patient, doing the genome editing in the lab, and then reintroducing the edited cells back into the patient through what is effectively a bone marrow transplant. So it involves multiple weeks in the hospital, and it's very expensive and it's very invasive, of course. And so many patients that could benefit from this aren't going to be able to do so because of the cost and the process that's involved. So I think we have a really interesting opportunity right now to change the way that this therapy is being delivered. And that's one of the reasons I founded the Innovative Genomics Institute, which is on my banner here, because we really want to focus on the ways that we're going to be able to bring this kind of therapy, and not only for sickle, but in other diseases as well, to a point where it's much, much more widely available for people to access it.